Antibiotic-resistant bacterial infections are a critical rising global health threat, projected to cause 39 million deaths by 2050. This resistance renders traditional antibiotic treatments ineffective, complicating the management of the infection, causing higher mortality rates. The impacts of these bacteria are already apparent. You may recall the E. coli outbreak linked to onions served at McDonald's just last October, which led to 104 cases. Another familiar story is the contaminated lettuce in Chipotle caused by a similar E. coli strain that caused over 1,000 cases of foodborne illness. With a clear demand for alternative treatments for antibiotic-resistant bacteria, there has been a renewed interest in phage therapy. This therapy utilizes bacteriophages, or phages, viruses that can infect bacteria but not human cells to specifically target and neutralize pathogenic bacteria. Phages are promising in mitigating infections because of their host specificity and self-propagation. However, an advantage and weakness of phage therapy is the specificity of the phages to infect and eliminate a relatively narrow range of bacteria. This becomes a challenge and leads to the need for multiple phages to treat complex multi-species infections. This study tackles this challenge by characterizing a bacteriophage that can target multiple species of bacteria isolated from a recurrent urinary tract infection. Our lab received 3 isolates from a UTI patient at UCI Health. There were 2 E. coli isolates and 1 Klebsiella isolate. We hunted for phages using wastewater from the city of Escondido. Wastewater is an excellent composite sample rich in microbes including phages. From our hunt, we returned 3 phages, Leo and Tot, which were E. coli phages, and Albert, which is a Klebsiella phage. Then, to enhance their activity against their hosts, we ran a directed evolution experiment, exposing the phages to a co-culture of the hosts. Essentially, we incubated 1 phage with 3 hosts over 5 generations of the phage. Interestingly, after this, Tot's host range expanded from just the 2 E. coli hosts to also include the Klebsiella. This is a fascinating result as cross-species infection of phages is extremely rare, and observing this phenomena after some directed evolution experiments highlights the potential for using the technique to expand phage host ranges to overcome the challenge of the narrow host specificity. My final presentation will include an in-depth characterization of 3 Tot 24.